Okay, this video is in answer to the very last question from our Zoom session that involved gene linkages. So the first piece that you have to remember in terms of genes and chromosomes and genetics is that genes are segments of chromosomes. Mendel's law of independent assortment states that two different genes assort independently of each other. And that's true if the genes are located on different chromosomes. If genes are not located on different chromosomes, if they are located on the same chromosome, they will be inherited together and not independently. So, what does that mean? First, let's review Mendel's law of independent assortment. Mendel's law of independent assortment means that if you have parents that are heterozygous for two different traits, each trait is inherited independently of the other. So if we have two parents that are heterozygous for P shape, round, and P color, yellow, but they're heterozygous, so they carry alleles for the wrinkled shape and the green color. That means that the probability of inheriting P shape is independent of the probability of inheriting color, which means that it is equally likely for this parent to give both dominant alleles or one dominant and one recessive allele to their offspring. Meaning we set up the Punnett square with 16 boxes, one being both dominant alleles, one being dominant for shape, recessive for color, one being recessive for color, dominant, recessive for shape, dominant for color, and recessive for both. Fill out the Punnett square, and if both parents are heterozygous for both traits, you can expect what we call our typical 9:3 to 3:1 phenotype ratio. Remember, phenotype is what they look like. And what that means is if both parents are heterozygous for both traits and the traits are on different chromosomes, then there is a 9 out of 16 chance for the offspring to show both dominant phenotypes, a 3 out of 16 chance for them to show dominant and for one and recessive for the other, and a 1 out of 16 chance for the offspring to show both recessive traits. This is true if the alleles are not linked, if they are located on different chromosomes. This is what independent assortment means. Now you're going to do a practice doing a test cross. Remember that we used this example in class many months ago, that in mice, being able to run in a straight line is a dominant allele, that having black fur also a dominant allele, that not being able to run in a straight line but going in circles, that's called waltzing, is recessive, and brown fur color is recessive. So if we were to cross a heterozygous running, heterozygous black mouse with a waltzing brown mouse, what I want you to do on a separate piece of paper is to do the Punnett square. Draw out the 16 box Punnett square with these two parental genotypes. Fill out the top, fill out the side. Take a picture of your completed Punnett square and upload it to this Google assignment so that I can see you remember how to do it. Don't peek at the key, but the answer is coming on the next slide. The way to set this up, and it doesn't matter which side you put which parent, but I put the heterozygous parent on the top. One possibility is both dominant alleles. One possibility, dominant for running, recessive for fur color. Recessive waltzing and dominant for fur color, recessive for both. That's this parent. Because this parent is homozygous recessive for both traits, there's really only one possibility. Little r, little b, the two recessive alleles are the only possibilities. What we wind up with a phenotype ratio, if these alleles are not linked, is 1/4 running black, 1/4 running brown, 1/4 waltzing black, 1/4 waltzing brown. If these genes are not linked, you would expect to get a 1-to-1 to 1-to-1 ratio for each phenotype, the same amount. This is a test cross of a heterozygote being crossed with a known homozygous recessive. So how do we determine gene linkage? We determine gene linkage by doing this test cross and looking at the phenotypes of the resulting offspring. Why does this work? In regular independent assortment, if genes are on two different chromosomes, so let's say gene A is on this chromosome and it's the dominant gene A, and gene B is on this chromosome and it's the dominant gene B, the potential gametes will have both dominant alleles or both recessive alleles. But because of independent assortment in metaphase, this orientation in metaphase 1 is equally likely as this orientation. This orientation will give us recombination, one dominant and one recessive allele in the gametes, so that when these gametes combine, we have an equal chance of having dominant and recessive alleles mixed together. Here are our 4 possibilities in our Punnett square: big A, big B, little b, big A, little b, little a, big B. Those are our 4 possibilities. So crossing any of these possibilities will get us all of those different possibilities of phenotype. However, if these genes are located on the same chromosome, then the dominant alleles and the recessive alleles will always be together, and there is no way to have a gamete that has little a and big B, or big A and little b, unless crossing over happens. And because crossing over doesn't happen 100% of the time, because crossing over will only happen if there's enough space between the two linked genes. The further apart they are, the more likely crossing over is to happen. The closer together they are, the less likely crossing over is going to happen. So the ultimate way of checking as to whether or not you're looking at gene linkage is if you see that the results of a test cross of one heterozygous parent and one homozygous recessive parent are 4 phenotypes that are not a 1-to-1 to 1-to-1 ratio, then the genes are linked, and any offspring that do not have the exact same genotype and phenotype of the parent as the parents are due to crossing over. Here's how I described that